Class, welcome back. Chapter 23, the Parasites of Medical Importance. I'll put a small disclaimer in at the beginning of this lecture. This is the first chapter where we're going to really dive deep into a particular category of eukaryotic microorganisms responsible for disease. So we're going to really start officially today with the organisms that are termed the parasites. The ones that are responsible for feeding and living off of a host while causing damage and destruction. And I will say today, I wish that I could be in the classroom with you watching your facial expressions as we go through this, because I guarantee you that probably by the midway through this lecture today, you will have a few of the ew faces and the oh my god discussions as you go through this. So get back, make sure you're not eating while you watch this, because you're going to see some pretty interesting things today as we look through diseases. So here we go, Chapter 23, Parasites of Medical Importance. Again, when we talk about parasitology, we're going to focus mainly on two categories of eukaryotes today. The protozoans, which are those unicellular-like organisms, and the helminths, or the parasitic worms. And these two groups are responsible for about 20% of all infectious diseases. Obviously, in countries that are well developed, have treated drinking water sources and are prevalent for having treatments and preventative measures for microbial diseases, these diseases are a lot less common. So as we said, the protozoans are unicellular, and most have some form of motility. Remember, one of the big traits about the protists were that they possessed a appendage for locomotion, such as flagella or cilia or pseudopods. And there's about 100,000 different species that fall into this protozoan category. Only about 25 of them are truly unique pathogens. And we're going to see that there's real key differences in the life cycles that we'll discuss today between many of these microbes. Most will have the actively feeding, metabolically growing state known as the trophozoite. However, some have that dormant, resistive structure that they're able to produce called a cyst. And we're going to see connections to how the cyst permits the microbe to be able to travel throughout the body. And we'll see that others have that they serve as kind of biological vectors, where the asexual and sexual aphases are occurring throughout the life cycle of the pathogen and the vector in which they're carried. So this is just an overview, table 23.1 in your book. Some of the key microbes that we are going to go through today, looking primarily at the body region impacted, as well as the reservoir, the particular source in the natural environment in which they can be found and originate from. Treatments, again, these are not going to be treated by antibiotics. When we talk about antibiotics, we're looking specifically at the bacteria. There are other classes of antimicrobial drugs, of which you'll see many of the drugs of choice here for a lot of these microbes. Things like chloroquine we hear about a lot with Plasmodium, which causes malaria. And, we talk about pyrimethamine, which is another one that's responsible for treatment of Toxoplasma, which causes Toxoplasmosis. So we're going to dive in first and start talking about the protists today. And then we'll move into the helminths, the parasitic worms, and finish up with a very brief discussion of the arthropod vectors that are responsible for carrying a lot of different diseases. So Entamoeba histolytica is the name of the protozoan. It is an amoeba, and remember that these amoebas have these projections known as pseudopods, which allow for them to be able to transfer or move from one location to another. And key about these is that they alternate forms. So they have the trophozoite, which is that actively growing form, and they can also produce a cyst, which is the smaller metabolically inactive, dormant form of the protist. These trophozoites have a extremely large nucleus, and however, they are going to lack most other organelles that we would think of within a eukaryotic cell. Humans are the primary host of the disease, and the way that this works is the cysts are swallowed, they're ingested through the mouth. They are going to be able to pass through the stomach. Now remember, the reason why the cysts are not destroyed is they are that dormant form. It's the protective structure to ensure that the pathogen is protected from the defenses of the body. And the human stomach has an extremely low acidic pH environment. So most things would be destroyed through passing through the stomach, but because the cyst form is there, it protects it from destruction. Those cysts are then able to enter into the intestines, where they're able to attach, they're able to release the actively growing form known as the trophozoite, and that trophozoite is then going to feed off of the tissues in the intestines. And it's going to result in a disease known as Amebiasis. And one of the key things is, in some cases, the amoeba can actually secrete cytotoxic enzymes, these enzymes that are able to destroy cells and dissolve tissues. And there's a slew of symptoms that result in amebiasis. Bloody diarrhea, which gives this kind of bloody and stringy diarrhea. It is also able to produce intense abdominal pain. We're going to see a lot of these diseases not only give you the intestinal symptoms, but you're also going to get fever and pain associated with them. And weight loss. Obviously, if you're releasing large amounts of excrement from the body, that is going to ultimately shift your body weight as well. The Entamoeba histolytica can be life-threatening, and that's because it's able to invade body regions like the liver and the lung and promote these tumor-like growths, which are called amebomas. There is about a 10% fatality rate, and the most effective treatments are things like metronidazole and chloroquine for treating Entamoeba histolytica. It's also well known that Entamoeba histolytica can also be transmitted in some forms through sexual contact as well. Now we're going to get in and start talking about amoebic infections of the brain, of which we have two. The first one is called Naegleria fowleri, and it's acquired through standing water. And then we have Acanthamoeba, which is different from Naegleria fowleri because it can either enter through broken skin or through the conjunctiva as well. So we could see things going, Naegleria fowleri is usually up the nasal passages. Acanthamoeba can either be through the broken skin or through the conjunctiva. And the end result is the infiltration of the brain, resulting in a meningoencephalitis, where there's an inflammation of the not only the spinal cord, but the meningeal coating that surrounds the brain tissue. Extremely fatal. We now go on to the ciliates. And for the big one that we're going to talk about, the first one is Balantidium coli. And we're going to see again that the reason they're called ciliates is because the surface of the cell is surrounded in these small hair-like projections known as cilia. The Balantidium coli is actually acquired through ingestion of the cysts, either in food or water. And it's usually found as a source in the animal kingdom, such as pigs and cattle. Once the cysts get in, the trophozoite is then going to emerge, and it's going to begin to erode the intestine, leading to the symptoms of pain, diarrhea, and the other ones that we would normally expect to see. The Trichomonas species is the first group of the flagellated protozoans that we are going to talk about. And this is a pear-shaped, kind of has a very narrow, pointy end that then widens out into this pear-shaped formation. It has a tuft of 4 flagella located at one pole of the cell, and also this undulating or kind of moving membrane. There is no cyst form with Trichomonas. It is only in the trophozoite or actively metabolizing form. The big form that we're going to focus on is this first one here, Trichomonas vaginalis. As we mentioned, Trichomonas vaginalis is an STI. It causes a disease known as Trichomoniasis, and it is a strict parasite. About 3 million cases in the U.S. every year. And in females, it produces a condition known as vaginitis, where you get this frothy, foul-smelling discharge that is produced from the vaginal tract. In males, it's usually a inflammation of the urethra known as urethritis, with a milky discharge that is produced. And metronidazole is the big target treatment for this protozoan. Giardia lamblia, which produces Giardiasis, is one of the most common waterborne pathogens that we know of. And it is very similar to what we saw earlier with Entamoeba histolytica, where the cysts of this particular organism enter through the mouth, through ingesting contaminated food. It has this kind of concave texture to it. And the reservoirs, again, are animals, things like beavers, cattle, coyotes. And the cysts can actually go out and survive in the external environment for up to 2 months. So these are incredibly hardy organisms. Again, ingested with food or water, very low infectious dose. Remember, we said the lower the infectious dose, the more prevalent infection is to happen, because it takes very few cells to cause that infection. So I want to talk about the pathway here. Again, the cysts are going to enter the mouth through food or water. The cysts are then going to travel through the stomach, able to survive the acidic environment, and travel to the small intestine. Once they reach the small intestine, the cysts will release the trophozoites, the actively growing form, and the trophozoites will begin to multiply and cause disease. However, in cases where dehydration in that intestinal environment occurs, the trophozoites will then go back into the dormant cyst form, and you can either see the cysts or, in cases, the trophozoites be released in the feces. So one of the key characteristic means of identifying a patient is to examine the fecal matter for the presence of flagellated protozoa. And again, we're seeing metronidazole as the major treatment that seems to be one of the most common ones so far. I will also mention that we can also kill the microbe besides the treatments. Boiling, the use of ozone, which is a variant of oxygen, and iodine are able to destroy the cysts of Giardia. The hemoflagellates are blood parasites, and we're going to get in and start talking about many of these obligate parasites that feed off the blood and the tissues and are life-threatening. And these are zoonoses, meaning that they are acquired from contact with animals or animal products. Found mostly in tropical regions and are transmitted by blood-sucking insects, they have incredibly complicated life cycles to them. So this is a very important slide to make sure that you know and are aware of the different stages of the hemoflagellates. So first of all, they have 4 different possible forms. The amastigote form means that it lacks a flagellum. The promastigote form is the form that has a single polar flagella, as you'll see down here. So amastigote, no flagella. The promastigote has the single anterior flagella. The epimastigote is the fully flagellated stage. And the trichomastigote is where we have the large kind of fully formed stage of this protozoan. So we're going to talk about a couple different types now, Leishmania and Trypanosoma, both brucei and cruzi. So the Trypanosoma species are distinguished by their infective stage. And they have the trichomastigote, which is that really elongated, kind of spindle-like shape to them, that fully formed stage. And there's 2 different variants. As I mentioned, there's Trypanosoma brucei, which is responsible for a disease known as African sleeping sickness, and Trypanosoma cruzii, which is known as Chagas disease. And each of these has a specific arthropod vector that's responsible for carrying the disease from one location to another. So let's start first about Trypanosoma brucei, which causes the African sleeping sickness. This is transmitted by a vector known as the tsetse fly. And there's actually 2 variants of Trypanosoma brucei. We have the Gambiense strain, which is endemic to West Africa, and the Rhodensiense strain, which is endemic to East Africa. And really, the way it works is this tsetse fly bites and takes a blood meal off of the host, which then injects the trichomastigotes, that fully developed form, into the blood. And it is then able to travel to body regions like the spleen and the lymph nodes and the brain, where you get things like damage to the organs as well as effects to the central nervous system. So again, we talked about this central nervous system effect. Some of the things that you're going to get are sleep disturbances, tremors, paralysis, coma, and eventually death. And that's why it's known as African sleeping sickness, because of the symptoms that we see with the tremors and the sleep disturbances. Trypanosoma cruzii, on the other hand, its vector is this little tick-looking bug known as the regivid or kissing bug. And the reason why it's known as the kissing bug is it actually bites around the opening of the mouth, usually to the sides of the cheek or the lip region, where it takes its blood meal. And really, here's the gross part, folks. The way that infection is transmitted is it actually excretes its feces into the wound after it takes its blood meal. And what you end up with are fever and swelling of the lymph nodes, as well as damage to the liver. You're going to see heart muscle and the large intestine are going to harbor that amastigote form. And remember, the amastigotes are the form that lack the flagella. So because it's in places like the heart, the intestine, and the brain, you're going to get very large inflammation of those key organs. We also mentioned Leishmania. And Leishmania causes a disease known as Leishmaniasis. And it's zoonotic, and it's transmitted by a vector known as the sandfly that takes a blood meal and is able to produce eggs after the blood meal. And it's endemic in these very temperate tropical regions. So what it does is it injects the promastigotes during the blood meal. Once they're in the body, they then begin to convert to the amastigote form, the form that lacks the flagella, and they multiply. So if macrophages do not come in and localize the infection, it's then able to become systemic and travel throughout the body. Leishmania can result in a whole slew of different disease conditions, such as this boil here that you see. This is known as the Baghdad boil, this ulcerative sore that forms on the skin. There's also a condition known as spondia, which is a result of damage to the skin and mucous membranes. And then systemically, it's able to produce things like fever, enlarged organs, and we have this variant version known as Kala Azar. And Kala Azar, again, is extremely fatal. And death actually results from things like anemia and destruction to the blood-forming tissues. So this is actually a form of Leishmania. We'll also talk about the Apicomplexans. And the Apicomplexans lack motility. So these do not have locomotor structures. And they're able to alternate between a sexual and an asexual phase. So they have very complicated life cycles. And the three big ones that we'll talk about are Plasmodium, which causes malaria, Toxoplasmosis, which causes Toxoplasma, which causes Toxoplasmosis, and Cryptosporidium, which causes Cryptosporidiosis. And these are transmitted either through food or water, or in some cases through the transmission by an arthropod vector, like we'll see with Plasmodium, which is transmitted by the Anopheles mosquito. So Plasmodium is the agent of malaria, and it is a sporozoan. The most fatal form is this one here, Plasmodium falciparum, is the most common, most fatal form of the 5 different species, transmitted by the Anopheles mosquito. There is over 300 million new cases each year, resulting in about 2 million deaths. And you'll see kind of the distribution as this equatorial region is where this is most common. The complex life cycle has 2 phases, and we're going to break these down. We have the asexual phase, which occurs inside the human host. So this is a biological vector. The life cycle occurs within different phases of the host. So the female mosquito is going to inject during its blood meal a form known as the sporozoite form. And that sporozoite form is then going to end up getting transmitted to the liver. And once it's in the liver, it undergoes a process known as schizogony, which is going to generate another form known as the merozoites. And it's these merozoites that are able to infect the red blood cells and circulate for between 5 to 16 days, depending on the species. The merozoites, as I mentioned, attach to the red blood cells, and they actually convert to the trophozoite, the actively growing form, and they multiply within the red blood cells, releasing more merozoites that become gametes or the sex cells. So right now, the reason why this is known as the asexual phase is this sporozoite, once it's injected into the blood and it gets to the liver, it does not need fusion of a sperm and egg to divide and create this new form, the merozoites, which then are able to infect the red blood cells and convert to the trophozoite form. The gametes are what are going to go on and go into the final phase, which is the sexual phase, the gametocyte phase, where the mosquito is now going to come back and it's going to take a blood meal. And when it does, it's going to withdraw some of those infected red blood cells, and those gametes are going to create sporozoites again in the stomach of the mosquito. Now, what happens is the sporozoites, as they come up, they actually block the salivary glands of the mosquito. And the mosquito gets this very rapid, rampant appetite. So they now start to try to take a blood meal to unblock those salivary glands. And as they take different blood meals on different hosts, they continuously infect the new patients with the sporozoite form, and this process repeats. So again, mosquito bites, injects the sporozoites into the human host. They undergo a division known as schizogony, producing merozoites. The merozoites then enter into the red blood cells of the liver, convert to a trophozoite form, which is going to release more merozoites. These will become the gametes that then the mosquito takes the blood meal again, withdraws those gametes from the red blood cells, forming more sporozoites, which are repeating that process again. So how do we diagnose malaria? Well, we look for the presence of the trophozoites inside the red blood cells on microscope slides in blood smears. Patients who have malaria also have these 48 to 72 hour cycles that produce these patterns of chills, fever, and sweating. And the most current-based treatment is the use of quinine drugs such as chloroquine, and there is a artemisinin-based combination therapy known as ACT that is also utilized. Vaccination is extremely difficult to develop because the pathogen can actually shift its molecules on the outside of its cell. They call that shifting. Toxoplasma gondii, which results in Toxoplasmosis, is another Apicomplexan parasite, and it lives naturally in the intestinal tract of cats. And these oocysts that form in the GI tract can either be transmitted through ingesting or inhaling the oocysts from the cat feces, or the cysts can actually be ingested from raw or undercooked meats as well. Most cases go unnoticed. So if you're not immunocompromised, most times the symptoms are going to present as things like sore throat, low-grade fever, and swollen lymph nodes. It's when patients are either pregnant or immunocompromised where congenital effects to the baby or damage to the brain and heart can occur in those hosts. I also want to talk about sarcocystis and the disease known as sarcocystosis. And when we talk about sarcocystis, one of the big things that I do want to get into is that these are parasites of cattle, swine, and sheep. So these domestic animals are the intermediate hosts, and they actually acquire the cysts while they feed. Humans are infected when they consume the meat. So when they consume the meat products, this is how they acquire the disease. Sarcocystosis includes the traditional symptoms, diarrhea, nausea, and the resulting abdominal pain from infection by the organism. And there's currently no specific treatment available. Cryptosporidium infects a variety of mammals, birds, and reptiles, and it results in a disease known as cryptosporidiosis. The organism exists in the tissue, also has that oocyst phase to it as well. The organism was really found in the early 1990s. There were about 370,000 cases in Wisconsin from contaminated water. And patients typically present with things like headache, sweats, vomiting, cramps, as well as diarrhea. They are able to penetrate those intestinal cells. AIDS patients can suffer since they're immunocompromised, a chronic persistent diarrhea as a result of infection by these pathogens. As we mentioned, it's an oral fecal transmission, so things like food and water, that's a big one, another waterborne organism. Those oocysts, as we mentioned, enter the small intestine, and they result in the symptoms that we've discussed: watery diarrhea, abdominal pain, fever. Babesia microti is the first protozoan to be tied to the bite from a vector such as a tick. And it is acquired from the bite from the hard-bodied tick, and it's a relatively rare zoonosis. The infection does resemble the symptoms of malaria, and again, it's diagnosed usually by looking at the blood cells in a blood smear under the microscope. Finally, we're going to get in and start taking a look at some of the helminthic parasites. And remember that these are the parasitic worms. And the parasitic worms are the most complex multicellular eukaryotic microorganisms that we know of. And we mentioned in our survey chapter, in chapter 5, many of these have very primitive organ systems and nervous systems that allow them to function with their environment. Some of the worms have separate sexes. Some are hermaphrodites, meaning that they contain both sex organs on the same animal. The adulthood and mating occur in the definitive host stage. The larval development occurs in the intermediate host. The transport host, or the vector, doesn't experience any symptoms from hosting the parasite. So here's an overview, table 23.4, of many of the organisms that we are going to talk about today, as well as how they are spread to humans. So let's pop in first and start talking about how the worms cause disease. Well, first of all, they are strict parasites. So they are going to attach to and feed on your tissues and body fluids. And as they do this, they are going to reproduce, and there's going to be an accumulation of worm and worm products at those particular body sites. So they are going to excrete in your tissues, they are going to lay eggs in the tissues, and diagnosis is really based on looking at a blood cell count, going in and examining tissues for presence of the eggs, the larvae, or the adult worms in the feces, the sputum, blood, and tissue biopsies. And there are a class of anti-helminthic drugs that are able to disrupt their metabolic processes, causing them to die off. They can also stop the worm from moving or from it attaching to different parts of the body as well. Again, table 23.5 has an overview of many of the drugs that are in this anti-helminthic category and the particular parasitic worm diseases that they're used in. So we're going to get in and start talking about the three forms of the worms: the roundworms and the flatworms, and tapeworms as well. So the nematodes, or the roundworms, are the most abundant, about 50 species that infect humans. They have this cylindrical body shape to them. They do have a complete digestive tract, and they carry separate sexes. So each worm is either a male or a female. And these are divided either into tissue or intestinal roundworm diseases. So we have two big groups that we're going to talk about and some of the representative organisms. So our intestinal nematodes include things like Ascaris lumbricoides, Trichuris trichuria, Enterobus vermicularis. The hookworms, we'll talk about Strongyloides stercularis, Trichinella spiralis, and then on our tissue nematodes, we have Wucheria bancrofti, Onchocerca volvulus, and Draculunculus medinensis. So we're going to get in and we'll start talking about the Ascaris disease first. Again, we mentioned this is a roundworm, prevalent mostly in the southeastern states, and is really humans are the only host. Ascaris can spread to humans through food, drink, or other contaminated objects, and it really has kind of a disgusting life cycle to it. The eggs are ingested. They hatch into larvae, and they actually burrow into the intestinal wall. They can enter into circulation and travel to places like the lungs and the pharynx. However, it's important to note that you can actually swallow this worm in two different forms. And what I mean by that is the larvae can actually migrate up to your pharynx. You feel like you have something crawling around in your throat, and you actually re-swallow them, and they return to your intestines, where they progress and develop to the adult form, where they are then able to come back up the pharynx and be swallowed down again. The adult worms actually lay about 200,000 eggs a day in your intestines. The eggs of this worm are spread throughout the food, and as we mentioned, the larva can actually get in and penetrate your lymphatic system and your lungs as well. Trichuria is known as whipworm. Humans, again, are the sole host of this microbe. The eggs are hatching in the intestine. About 3,000 to 5,000 eggs are laid daily. The worms can actually pierce your capillaries, causing severe hemorrhaging. And this can lead to things like dysentery and rectal prolapse, with the most fatal cases in children. Enterobius vermicularis is kind of known as the 2F's disease. It can be transmitted on the surface of the fingers and also from fomites or inanimate objects. And it produces this very prevalent disease known as pinworm. And its eggs, as we mentioned, are picked up from the surroundings and swallowed. Once they arrive in the small intestine, they then develop into the adult form. And I mentioned in Chapter 5, our last slide, we talked about the self-inoculation that occurs with this disease. So at night, the worms, mature females, actually emerge from the intestine to the opening of the anus to release eggs. And you get this anal itching that occurs, and the scratching of that itching actually causes the self-inoculation. The disease is identified through what's called the tape test, where they literally adhere a piece of tape to the tissue surrounding the anal opening and look for, after the tape is removed, the presence of these mature female worms or the eggs that have been deposited. The hookworms, and one of the big ones we're going to talk about is Necator. The hookworms have these oral cutting plates on the hooked mouths. They have these kind of curved mouths with these cutting plates that permit them to anchor to the intestines. And as we mentioned, Necator americanus and also Ankylostoma are the two big worms that fit into this category. The humans shed their eggs into the feces, and when they hatch into larvae, if they're on the ground and in the soil, the larvae, this filiform larvae, can actually penetrate through the skin on bare feet. So I bet you everybody's saying, oh my God, I'm not walking around barefoot outside anymore. Once the larvae enter the body, they can travel through the blood up to the lungs, and again enter the throat where they're swallowed. Those worms are going to mature in the intestine once they're swallowed and complete their life cycle. Blood loss is incredibly significant, resulting in anemia. However, you may also see digestive symptoms such as nausea, vomiting, and cramps associated with that swallowing of the worm in the throat back into the intestine. Strongyloides stercoralis, it produces a disease known as Strongyloidiasis. It's known as the threadworm, and these are tiny little roundworms that either have part of their life cycle in humans or the soil. Again, larvae can penetrate the skin, they migrate to the lungs, and can be reswallowed, allowing them to enter the intestine. They can reinfect the same host without leaving the body, and you're going to get diseases such as destruction of the blood cells, diarrhea, swelling of the liver. Trichinella spiralis, it causes a disease known as trichomoniasis, and trichinella is acquired usually from ingesting pork or bear meat. And what happens here is the larvae are encysted, meaning that they're covered in a cyst form to protect them. They're ingested from contamination of an undercooked meat product, such as pork or bear. When they're ingested, the cysts actually hatch and mature in the intestinal lining, and then the adults now reproduce and form new larvae that can enter into circulation. And the larvae actually form cysts in your skeletal muscle and can remain there for several years. All right, we're going to move into the tissue nematodes. And these tissue nematodes complete their life cycle in the blood, the lymph nodes, or the skin. And these have a series of elongated filamentous worms that are spread through arthropods, so some type of a vector. And they cause filariasis, which is a kind of disforming disease. So we have things like Autieri, which causes elephantiasis, and river blindness, and also eyeworm, which is known as loa loa. So the first one that we'll talk about is the elephantiasis, which causes massive swelling of the extremities because the infection blocks your lymphatic circulation. Spread by mosquitoes, this is an example of that swelling due to the blocking of the lymph nodes, resulting in the swelling of your extremities. The river blindness is transmitted through the black fly vector. The adult females can actually move through the blood and up to the eyes where inflammation occurs. And it can lead to what's known as river blindness because the worms have actually invaded your eyes. And then lastly is loa loa, which is a bite from a small fly. The worm can actually migrate under the skin and can even enter the eye like we saw with the river blindness. And then we have Dracunculus medinessens, which causes a disease known as Giardiasis. And it's caused by the guinea worm. The parasite is carried by Cyclops, which is a microscopic arthropod that's found in still water. The World Health Organization has caused cases to drop from about 3.5 million in 86 down to only about 24 cases. So this disease is almost fully eliminated. The trematodes, which are flatworms that have these flattened bodies, we are going to remind ourselves that these have the very primitive digestive, excretory, and reproductive systems. However, they lack circulatory and respiratory systems. And we're going to see that their hosts are usually animals such as snails or fish, with humans being the definitive host. So here again is our example of our trematode. First one we're going to talk about is the schistosomes, and the particular pathogen is known as schistosoma, and it results in a disease known as schistosomiasis, which is a very prominent parasitic disease from a fluke. And these flukes live in humans who will release the eggs into water. The larvae develop and are living within the intermediate host, which is a snail. Once the larvae are infected through the skin, they penetrate and move to the liver, and the adults actually migrate to the intestine or bladder where they shed their eggs, resulting in an enlargement of the organs. These worms are also known to penetrate your hair follicles, and they can actually pass in the bloodstream to the liver as well. We have the Clonorchus worm, which is able to cycle between mammals, snails, and fish. Humans become infected because they ingest the Cercariae form of the worm in inadequately cooked fish products. The Cercariae, or larvae, crawl into the bile ducts, mature, and shed eggs into feces. Fasciola hepatica has a cycle that alternates between herbivores, snails, and aquatic plants. Humans become infected by eating those aquatic plants, and the fluke actually lodges within the liver. You do not need to worry about this particular one. We will not discuss this one. The cestode or tapeworm infestations are also flatworms, but they have this long ribbon-like body. And the sections of the long ribbon-like body are known as the proglottids. And these little body segments actually contain the sex cells. These are hermaphroditic, meaning that they contain both sex cells on the same worm. And they have a head region known as the scolex, which has the hooks and suckers that allow them to attach to the intestinal tissue. So we're going to talk about Taenia saginata, which is known as the beef tapeworm. And these have extremely long bodies, up to 2,000 proglottid segments, humans being the definitive host for the worm. The Taenia saginata is acquired by animals as they graze on the land and are contaminated with human fecal matter. The infection for us occurs from eating raw beef, and the larvae can attach to your small intestine and become an adult. Abdominal pain and nausea result, and you can actually find the presence of those proglottid body segments present in the stool. Taenia solium, on the other hand, is the pork tapeworm. Again, by ingesting the cysts or the eggs are going to hatch in the intestine. They're going to release the tapeworm larvae that will migrate to tissues, and they can actually damage the heart, the eye, or the brain, causing things like seizures or psychiatric disturbances. And finally today, we'll just mention some of the main arthropod vectors responsible for transmitting diseases. Arthropods are named the way that they are because they are they have jointed legs and they have a hard exoskeleton. So things like the arachnids, spiders, and crustaceans fall into this arthropod category. And many feed on the blood and tissue of a host during their life cycle. And this allows them to transmit infectious microbes in the process of their feeding. So we have things like mosquitoes, fleas, and lice, which are all common arthropod vectors that are able to transmit microbes in the process of contact with a host. Ticks we see and we'll see a lot throughout the course, whether we're talking about protozoan diseases like Babesia, or bacterial diseases like Borrelia, which causes Lyme disease. So this will give you kind of table 23.6 is several slides long and walks through a lot of the diseases. Again, you don't need to memorize all these right now. We will talk about these as we go through the chapter. For the ones that we did talk about today, like the tsetse fly and the sandfly, the black fly, make sure that you're familiar with those vectors right now and which diseases have those vectors. So you'll see that we will talk about them in several different chapters throughout the semester. So as a rule of thumb, I want to go over, as you're starting to prepare and organize your notes, it is really important for you to list out, and I would create kind of a study guide or a note sheet of the different diseases that we have talked about today, with not only how they're transmitted, but I would also be familiar with their symptoms, and in some cases, what the causative agent is. So make sure that you're familiar with the disease-causative agent relationship, how the disease is transmitted, and how the disease causes symptoms within the host. Okay, and that will help you make sure that you have your notes kind of all prepared prior to studying for the exam. Again, if you want to run through any of these things or ask further questions, feel free to stop in at office hours on a weekly basis, and I will address your questions then, or send me an email, or even post a question to the instructor within the Blackboard discussion board, and I will answer the questions there. I hope everybody's doing well, and I look forward to seeing you again really soon. Take care.